Uh, it's always pressure to follow Professor Yakub, and I'm trying to explain here what we have been doing last 6 months in the QCRC, also last 2 years in the Imperial College. According to the talk, we are going to discuss here about how we are designing the scaffolds for tissue engineering, hot valves. You know that this nation has a serious problem of having hot valve diseases. It's a burden in this region, and it requires the collaborative efforts from QCRC and Heart Hospital. Of course, we can go with the living valves, but it comes with the homographed availability, and this gives a lot of major problems. So that's why we need to go for tissue engineering, for which we have to design the scaffolds which will give the base for the tissue engineering. So what is tissue engineering? Actually, why we are concentrating about the scaffolds? These scaffolds, you can see that they are basically providing the environment for the tissues, as for the cells, to arrange in a proper manner in the 3D space, and then they are providing not only the structural integrity and also providing the signals, as Professor said. So when you combine these cells with the scaffolds, they will direct the cells into the proper way of arrangement, and then we will get the tissue engineering construct. So we have a big responsibility to emulate the ECM, the extracellular matrix. This rod-like structure is called collagen. These are the basic scaffold material right now successfully used for tissue engineering. So these kind of rod-like structures are wrapping up the cells properly to give the proper structure. That way we can get proper tissue materials which can be used for many surgeries. So you can see there are different types of scaffolds. Collagen is a natural collagen is always having instability, and also because it is insoluble, so it has it's coming with some antigenic properties, and also it is not flexible. If you want to introduce some signals which will be important for inducing some factors on the cell surface, then you have to go with collagen mimetic peptide way of introducing the scaffolds. That's why we are concentrating these collagen mimetic peptide scaffolds for tissue engineering. This is the normal structure of collagen. You can see the macromolecular structure here, which is available on the extracellular matrix. But the building blocks for this structure is called collagen mimetic peptides. They are unique structures. They are called tropocollagen helix. If you concentrate on this collagen mimetic peptide, you can able to translate this structure into nanofiber structure after engineering whatever the desired properties you required for your specific tissue engineering. Before going to detail, we should look at the structure of collagen, because it is having so much of information inside. It looks like this is the nanofiber you see on the surface of extracellular matrix, but when you go deep and see the molecular structures, they are arranged in a proper way. This is the triple helix I am talking about, the collagen mimetic peptides. When they arranged in a proper way, in the staggered manner, they are producing these nanofibers. These collagen triple helixes are basically constructed with three important residues. These are repetitive, proline, hydroxyproline, and glycine. These are the building blocks for this collagen structure. So when you concentrate in the the atomic level and the residual level, then you can able to introduce and play with the sequences to introduce new signals into the structure, and then you can able to produce the macromolecular collagen. This is the advantage of using the mimetic collagen. We are trying to introduce different properties. We can increase the stability, and also we can introduce functionalize with our desired functions, desired signals, so that we can able to go through from the bottom up way, we can able to produce the collagens which will be very useful for the many purposes like therapeutic agents. And why we are starting with computational biology? I'm basically a computational biologist who are designing these collagens, who are trying to understand the structural properties, because most of the proteins structure determines the functions. So that's why we are trying to understand using the computational approaches how these structures can be modified, how we can enhance these properties, and then how we can functionalize this collagen. So that's why we started with bioengineering of CMPs. This is the first approach which will increase the structural stability and then biofunctionalize. So what we are trying here is to try to mimic the same collagen fiber properties in the small the same building blocks of collagen, so that we can able to mimic the same collagenous properties. At the same time, we can enhance the properties. And the next step, we are trying to combine this CMP, collagen mimetic peptide, with the peptide amplifies. These are the new emerging biomaterials which is also can be used for as a biomaterial. And also we have recent developments to attach these CMPs with synthetic nanofiber, which is also synthesized by our colleague in the ARI field. So what are the things we should consider when we reproduce the collagen? So these are the prescribed list of things we have to consider. It's not like you can just synthesize the small peptides and then they can be a collagen. So we have to consider there are several structural properties we should conserve. At the same time, these molecules should be remodelable, and then they have to be precise in size, because the synthesis is also the major concern. And then we have to program them to self-assemble by introducing proper charges. And what are the advantages? Why we need to start with the computational techniques? If you go to the laboratory, we have to start with, if you have at least hundreds of mutations, you have to test one by one. It will take lots of time. But what the computational techniques recently developed, last decades, it has been developed very well, and they are giving rapid tools, try to point out exactly where to start, so that you can save a lot of time and prioritize where to start. So these are the examples how you can approach this computational modeling. It is nowadays a part of the, because all the research is going in interdisciplinary way, so such a way you can see the structure function relationship to understand the structures from the function from the structures. These tools are majorly used. You can design the structure and then you can go for the molecular simulation, so that you can have an idea before going to laboratory, and then you can understand where they can interact with using the structures available in the protein data banks. And then you can also understand how these proteins or target proteins can interact with each other to do biological functions. So you can try to imagine how we are going to do this collagen mimetic peptide, bioengineering of collagen mimetic peptide. If you consider this is the normal way of building Eiffel Tower or any constructions. So the same way, but we are using at the ultra scale level. So we are going to do the molecular refinement and molecular interactions by the way of increasing the interaction between the molecules, and then we are going to produce it. So this is the starting point. When we started, we had collagen structure. This is the triple helical structure, two triple helical structures of collagen mimetic peptide. They had this kind of sequence inside. So what we did, we identified hotspots. Hotspots are important for the structural properties and also important for the function. So we didn't disturb this hotspot, but we mutated by introducing, we introduced few charged particles to enhance the interaction between the molecules, so that they can self-assemble, or they can the stability of the molecule can be increased. So by mutations we have introduced without affecting the hotspots, and then we introduced the cell binding motif, which can be used for the extracellular interactions. After that, you can see that the original peptide has been dissociating, but when we introduce the mutations, they can make interactions by the way of in hydrogen bonds, and they can stay stable in the particular environment. And then if you go with the other binding motif structure, it's also showing that the association is there because of the refinement by introducing mutations. This work was published last year, and it was accepted as a leading opinion in the Journal of Biomaterials. After that, we started getting more information. The theory was there, and we gave to the laboratory, and they have developed and they have tried to recreate the same structures. And then we got a few more ideas, so that we have refined the structure to get more stable, and also the lengthy wise we have to increase it, because the validation suggested that it is needed for the thermal stability. So then we started to remodel the structures, and then we got thermally stable two new versions of peptides. We found that why they got very good structure when we increased the length and adjusting the sequences a little bit. We found that the molecular structures, intramolecular interactions are very important. So when we increase or when we remodel the structure in a proper way, then we will get an increase in the interactions. So automatically intramolecular interactions affect the global structural stability. That's why we got the structures in very stable manner. It is next given back to the validation, and now we can see that circular dichorism spectroscopy is the technique which will identify whether the structure is forming triple helix or not. This is the indicative of triple helix formation. When the people doing the collagen study, they will identify if there is a peak at the 225 nanometer, they will get that this structure is forming triple helix. So it is validated at the experimental level, and the temperature you can see that nearly physiological temperature, it is stable, so we can be moving to the next level. And the other validation is also done by circular dichorism. You can see that the melting, when you introduce this kind of molecule in a tissue engineering practice or other surgeries, it should be stable inside the physiological condition. So that's why we have to check whether they melting in a proper. This is the way they'll identify. If they get this kind of sigmoidal shape curve, then they say that they are trying to be stable there, and it is the indicative of the stability. And the next step, we started the programming self-assembly at QCRC. So why we are trying to identify this kind of interactions? This is actually mapping the surface charges, because protein-protein interactions always depend on two major forces. One is hydrophilic and another one is hydrophobic. The hydrophilics, positive and negative charges are called hydrophilic forces, and hydrophobics are water repelling forces. So when we give the structures proper way of charged, and if you balance the structure with this kind of two hydrophobic and hydrophilic forces, they will automatically interact. When they interact, they will go for the function. So this is what happening in the biological system. So that's why we are trying to map first before going to detail and before going to design our experiments. So what we are now going to do, make the structures with the proper charges so that they can make the proper interaction, self-assemble, then go for the fiber, then these fibers are used for the tissue engineering. So this is the technique, this is the strategy we are trying to implement, and this is the already proposed theory. Here you can see that this is the CMP, the triple helical structure, but in between there is a long gap, and there is a big question how they are forming self-assembling to form this nanostructure. So that's why what we are doing here, we are trying to mimic this structure into a simplest model, and then trying to trace out how they are trying to step by step move to the fiber level. So you can see that, as I explained already, the hydrophobic, hydrophilic are the major forces. So we designed three simplest models, one with full of hydrophobic and one with full of hydrophilic, and then one with a combination of hydrophobic and hydrophilic. So when you do the simulations, we get this kind of structures. So we can see that the nanofibers kind of structures are forming when we introduce at proper concentration, when you give the proper temperature, pressure, and the concentration. So everything's giving everything in a place to make this happen. And also we are getting different results like this. This is also can be possible. So they can arrange in a different way, still they can make it as a collagen like fiber. So this is like a hollow tube like structure. They can directly form as a collagen like structure, or they can be directly forming as a long structure like fibers. But anyways, this experiments still need to be validated in the laboratory level, but we are getting good progress of getting collaborations to get this to be done very soon. So this is the very recent experiment I am doing it. So if you see, this is the starting level, and then the next level, you can see the aggregation is happening, and this is the third level, the stage 3. You can see that the aggregation is increasing, and the last level, you can see that the bed-like structure. Maybe it is trying to explain that it is trying to reach this kind of extracellular matrix kind of thing. So this can be reproducible when we have a proper way of handling all the properties. And the next level is to functionalize these CMPs. Only the structure cannot be made. They are not going to make sense, because we need to give the proper signals to the cells. So what we are trying here, these are the two other peptides. The peptide amplifies are available. So you can see that these peptide amplifies and collagen peptide amplifies are not very different. But I would like to introduce first what is peptide amplifies, what is the difference between the normal collagen and this peptide amplified nanofibers. You can see that the basic unit of the peptide amplifier is the hydrophobic and hydrophilic. This hydrophilic, not only the hydrophilic, the charged or signal residues, the signaling epitopes can be introduced here. You can see the same structure is reproduced here. This is the hydrophobic and this is the signaling part. So when they arrange together, because of this hydrophobicity, they can form a clear, long nanotube structures, and the epitopes are exposed on the surface of the nanotubes. But not only one or two epitopes, there are millions of epitopes, because the structure is designed like that. When you give the nanofiber full of charge, full of signals, then the cell surface proteins doesn't have any difficulty to interact with and take the signals what they want. So this signaling method will be very effective when we try to combine this collagen into this peptide amplifiers. So that's what already people have done this one very recently, but it is not specific for the tissue engineering. So you can see that this is the peptide amplifier section, and this is the triple helical collagen section. So they are trying to integrate this, and then they are trying to make the collagen peptide amplifiers, the new hybrid structure, which will be used for other purposes. They are designed for some other techniques, but now we are trying to develop this for tissue engineering at QCRC. And this is the first step towards that. We are trying to understand, we already completed this work by understanding the mechanical properties of this structure, the peptide amplifier structure. So now we have a clue that how we can improve the mechanical property, and the next step we are going to target to introduce very vital signals on the top of the structure, so that we can able to enhance the signaling property and functionalize. And the next step the next approach we are trying as an alternative approach for what we have done earlier, to increase to functionalize the nanofibers. Our colleague Jerome is working beautifully on these structures to create this kind of nanofibers. So what we are now trying here is to functionalize this peptide by attaching some CMPs on the surface of these nanofibers, so that these synthetic nanofibers can be converted into biologically very valuable structure. So I would like to summarize the molecular modeling is valuable to before going to laboratory, because it can fine-tune the structural and functional components, especially in the CMP case. It is a very unique but complex structure. So this kind of molecular tools are very useful. So that's why we are applying and getting a very productive results. And we are trying to improve the program self-assembly property, so that we don't need to be focusing on that. If you place the charges and if you give the proper environment, then they will automatically self-assemble, and then they can form nanofibers. And also the functionalization, as I mentioned, it is very important, because without signals you are not going to pass anything from extracellular to intracellular. So it is also important. We are customizing by getting more information from extracellular matrix proteins like integrins. And also we are trying to use the new strategy of developing these peptide amplifiers into collagen peptide amplifiers specific for the tissue engineering. And I would like to thank the organization, especially QCRC, for giving a lot of support. And also I would like to thank colleagues Muthu, who is doing synthetic work in the Imperial College London with the help of Tony Cass, and Jerome, who did the synthetic nanofibers, and Adrian for the discussions, and Sophia and Professor Magdi for supporting all over the work. Thank you very much.
Thank you, Navaneeth, for the presentation. Congratulations for this excellent, and I guess for me it's amazing work and results. I guess probably this is the first maybe collaboration between QCRC and the Imperial College. At least there are some results and outcome out of the collaboration. I know we're looking forward for the other research projects, but I think this is a step forward and promising work. Thank you for that. So the floor is open for questions and comments.
Thanks for taking the presentation. There was a cartoon in The Economist about 6 or 7 years ago, presenting a man wearing a mask, watering a pot with a tree shaped like a heart, and the branches in the form of the great arteries. And this, the title or the headline of this cartoon, organs grown to order around the corner. Are we still looking for the organs to be grown according to order? Is the corner nearby, or the corner is still miles away?
I think we are trying to reach step by step, but I think it will take some time.
I mean, as I said, the title was, near the organs grown to order, and that was near the corner about 6 years ago. And now the 6 years have gone, and still we don't know whether we are near or far away. And when will we see the first grown to order valve?
Please.
Can I step in?
Yes, please.
Thank you. I think it's an excellent question. The thing is to do it in stepwise and not grow the whole organs, for example, like the whole heart. It might happen, but to grow specific parts like a valve, a vessel, a pacemaker, and then put it all together. I think with valves, we're very near. What is very near? we've been saying that for some time. I think it's 2 to 3 years, and we're starting now with having patches and valves with living cells, and that can be used as patches and as valves. So what is around the corner, I think it's about 2 to 3 years for small parts of the organ. When is the whole organ can be produced to order, like you say, that might take a bit longer. And experience has shown anyway that when we say 10 years nowadays, it usually is shorter rather than longer because of how research is accelerating in a fantastic way, like knowledge accumulating in the last 2 years is equal to knowledge accumulating in the previous 10 years, etc. So, very hopeful.
And if you have the mechanism to stimulate the cells to grow, I mean, as a valve or something, do you have really something to prevent them from overdoing it or underdoing it?
Brilliant question again. Shall I answer that again? Yeah, I think when you stimulate cells, you're always very, I mean, that's not this work, by the way, because he's giving signals with functionalizing the construct, but never putting oncogenic things. But if you use IPS, for example, then if you use a molecule to stimulate them grow, then you have very high risk of having a malignancy. But the way to do this is to stimulate them for a week, and you have the means of switching off the stimulus at the specific time, not to allow it to go on and on. Obviously, I'm talking on theoretical grounds.
Thank you, Navaneeth, for this informative presentation. I have 2 questions. I had one concern regarding the cytotoxicity of CMP. Have you any idea if there is any in vitro or experimental tests that have been made to look for the cytotoxicity of CMP? And the other question is, you think in the future the biomedical engineering or tissue engineering can make a kind of scaffold that have a signaling role? Or is any way to have a signaling synthetic scaffold?
Yeah, it's 2 important questions. Actually, the first one, I'm not expert in cellular study, but my colleague, Dr. Ivan, did a few experiments with CMPs, what we have designed, but they identified that it doesn't have any kind of cytotoxicity according to our structure. The next question you asked, we are trying our best to give the proper signals. We believe that extracellular matrix, only the collagen has more potential to give the proper signals that will be very helpful for the proliferation and also for many other purposes. So that's why we are sticking to the collagen to go to the excellent scaffold preparation, especially for tissue engineering.